Hello, my name is Joshua Mugabi. I welcome you to Cell Chemistry Self-Assessment. Apart from the food, what other substances do cells need to take in? Apart from the food, what other substances do cells need to take in? Cells need to take in water and salts in addition to food. They need to take in water and salts in addition to food. Water has a high capacity for heat, that is thermal capacity. This is an advantage in living cells because select one of the following statements. A. Any rise in temperature is small in comparison with the amount of heat absorbed by a cell. B. Any rise in temperature is large in comparison with the amount of heat absorbed by a cell. C. The 75% water in a cell does not retain a lot of heat. D. Any change in temperature will cause a cell to heat up or cool down quickly. So we are saying water has a high capacity for heat, that is thermal capacity. This is an advantage in living cells because a high thermal capacity means that any temperature rise is small in comparison with the amount of heat absorbed. This helps to protect the cell against extremes of temperature. A. Give 3 examples of cell structures which contain structure proteins. B. What is the other type of protein in a cell? A. We are saying give 3 examples of cell structures which contain structure protein. So we have the cytoplasm, the cell membrane, membrane systems in the cells, the nucleus, and mitochondria all contain structure proteins. Then part B, we asked, what is the other type of protein in a cell? And the answer is enzymes are the other types of cell proteins that can be found in a cell. Name the chemical elements present in a protein. Name the chemical elements present in a protein. Protein contains the elements carbon, hydrogen, oxygen, nitrogen, and sulfur. What name is given to the subunits which make up all proteins? What name is given to the subunits which make up all proteins? All proteins are composed of subunits called amino acids. They are composed of units called amino acids. A protein molecule which is denatured has A. Split into smaller molecules. B. Changed its shape. C. Combined with other molecules. D. Been diluted. A protein which is denatured has changed its shape. What kind of substance is a lipid? What kind of substance is a lipid? A lipid is a fat or oil. It may be combined with other substances like phospholipids or lipoproteins. In a cell, where are lipids found? In a cell, where are lipids found? Lipids are found in cell membranes and other membrane systems in the cell. Some cells may have food reserves in form of lipid droplets. A. What are the two types of chemical compounds which combine to form a lipid? B. What elements are present in a lipid? A lipid or lipids are formed from the combination of fatty acids and glycerols. Then when we talk of the elements that are present in a lipids, lipids contain elements carbon, hydrogen, and oxygen. Name 4 examples of compounds which are classified as carbohydrates. Name 4 examples of compounds which are classified as carbohydrates. Part B. What elements are present in carbohydrates? Examples of compounds classified as carbohydrates, we have sugars, for example, glucose, fructose, maltose, sucrose. We have starch, glycogen, and cellulose are all examples of carbohydrates. Then part B, we're asking what elements are present in carbohydrates. Carbohydrates contain elements carbon, hydrogen, and oxygen. Write the formula for glucose. Write the formula for glucose. The formula for glucose is C6H12 or 6. C6H12 or 6. If this structure or symbol represents a glucose molecule, draw A, maltose molecule, and B, part of the starch molecule. Maltose is a disaccharide made up of 2 glucose molecules. So the structure of maltose is having 2 glucose molecules combined together. And part B, starch molecule, is a polysaccharide made up of very many glucose molecules combined together to form part of the starch molecule. Select the most appropriate words from the list below to complete the following paragraph. All cells contain dash, which are dash, and act as dash, which dash chemical reactions. The reactions do not dash the dash, which can take part in further reaction. And the list we are choosing from, we have substances, proteins, enzymes, catalysts, speed up, use up, and slow down. Those are the words we are using to fill in the blank spaces. You can pause and first work it out. All cells contain enzymes, which are proteins, and act as catalysts, which speed up chemical reactions. The reaction does not use up the enzymes, which can take part in further reactions. Enzymes will usually react with only one substance. This can be explained by lock and key theory. If this theory is correct, which of the following substances represented by P, Q, R, and S would be acted on by enzyme N? So we have the substances P, we have Q, we have R, and S. Which of these substances will be acted on by enzyme A? You can pause and find out. Using the lock and key model, enzyme A is most likely to react with the substance R. So this enzyme A, if we are using the lock and key model, is likely to react with substance R, because that's where it is going to fit as a key to give us this intermediate. If an enzyme-controlled reaction normally takes place at 10 degrees Celsius, in general terms, how will the reaction be affected by A, a fall in the temperature to 20 degrees Celsius, B, a rise in temperature by 20 degrees Celsius, C, a rise in temperature by or to 65 degrees Celsius. If an enzyme normally works at 10 degrees Celsius, then part A, a fall in temperature to 2 degrees Celsius will slow down the reaction. And part B, a rise in temperature to 20 degrees Celsius will speed up the reaction by 2 times. And C, a rise in temperature to 65 degrees will denature the enzyme and stop it working, though the reaction may speed up at first. But later on, because of the increase in temperature, the enzymes are going to be denatured. If an enzyme is denatured, why does it no longer work? An enzyme which has been denatured has changed its shape and will no longer combine with its substrate. The substrate it acts on, meaning there is not going to be any enzyme substrate complex formed because the enzyme has changed its shape because it has been denatured. The graph shows the rate of an enzyme reaction at different level of acidity or alkalinity, pH. From the graph, what is the optimum pH for the enzyme, this enzyme? A, pH 2, B, pH 10, C, pH 7, or none of the above. This is a graph showing the reaction or rate of enzyme reaction at different alkalinity / acidity pH. At pH 2, we see this is the rate of reaction somewhere here. When it comes to pH 7, somewhere here. The rate of reaction is there. And when it comes to pH 10, you can see the rate of reaction is there. So which one is the optimum pH for this enzyme? The optimum pH is 7 because the rate of reaction is greatest at this pH. So when you look at pH 7 here, you will see that it's where we have the maximum rate of reaction. Therefore, it means that the optimum pH for this enzyme is pH 7. That's where it produces a lot of products. A protein digesting enzyme, when mixed with the starch solution, would A. have no action, B. produce glucose, C. produce amino acid, D. digest the starch. A protein digesting enzyme would have no effect on starch because for it digests proteins, does not digest starch. Therefore, it does not have a site that is conforming to starch substrate. Therefore, there would be no effect on starch solution. Select the most appropriate words from the list below to complete the following paragraph. All enzymes are produced inside dash. Enzymes which do their work outside cells are called dash. Enzymes which do their work inside cells are called dash. Most of our digestive enzymes are examples of dash enzyme. And the list we are to choose from contains animals, extracellular, intracellular, cells, digestive, nuclei, catalysts. You can pause and fill the paragraph. All enzymes are produced inside cells. Enzymes which do their work outside cells are called extracellular. Enzymes which do their work inside cells are called intracellular. Most of our digestive enzymes are examples of extracellular enzymes. Give 2 examples of chemical reactions which are catalyzed by enzymes in the course of brewing. Give 2 examples of chemical reactions which are catalyzed by enzymes in the course of brewing. In the course of brewing, enzymes in the grain catalyze the conversion of starch to maltose. Enzymes in yeast catalyze the conversion of maltose to alcohol. What does enzyme catalase do? Catalase speeds up the breakdown of hydrogen peroxide to water and oxygen. Substance A is being investigated to see if it is an enzyme. When substance A is mixed with substance B, a reaction takes place. A control experiment is conducted using a sample of A which has been boiled. Part A. Why is boiling used as a control? And part B. If the reaction still worked after A had been boiled, what might be your interpretation? Pause and try to figure out the answer. A. We asked, why is boiling used as a control? Boiling denatures enzymes. If a substance still works after boiling, it cannot be an enzyme. Then B, if the reaction still worked after A had been boiled, what might be your interpretation? If the reaction still worked after A had been boiled, either A is not an enzyme, or if it is not necessary for the reaction. In an investigation to compare the rates at which starch is broken down by an enzyme, A. What test is used? B. How do you know when the reaction is complete? What test is used? The test for starch is iodine solution, which goes blue. How do you know when a reaction is complete? When no blue color appears after adding iodine, all the starch has gone and the reaction is complete. Thank you very much for participating in the chemistry, cell chemistry self-assessment.